Do you think that's the only way to sort of see what's next is as far as what works and what doesn't work is trial and error?
Yes, we're trying in the lab. We're trying to figure out what makes them grow, and then we look for drugs that block the growth. It's just like cancers. The problem with AVMs is that they're all different. They're not the same. There's some focal ones which are just one localized, and there's multifocal like yours, which is just scattered multiple places. And those are very difficult to control. We think that those are more like tumors versus the others are more just blood vessels that, because they shunt from arteries to veins, it makes them get bigger and bigger. It's what we'll call a hemodynamic mechanism that makes them enlarge. But there are others that seem to invade the skin or invade bone or invade tongue, and they're very destructive. And we've shown a lot of similarities to tumors. We do these special tests, and they're testing very similar to tumors. The tumors will grow and multiply with cells, and that's the difference between some AVMs that are just blood vessels that keep getting bigger versus cellular growth. And that's the ones that we think drugs can have an effect on, are the ones that have cell growth or turnover. And then you try to block that turnover. With doxycycline, what we found is that AVMs have what's called MMP, which is a matrix metalloproteinase, which is an enzyme. That enzyme can actually break down tissue and allow blood vessels to grow into them. By using doxycycline, we think it can stop that process and stabilize it. So we're looking at the we'll call the pathophysiology of AVMs, and then trying to find blockers. That's what they're doing with cancers as well. And they're actually finding quite a few with cancers, but no one else is studying AVMs because they're so rare compared to cancers.